A sobering update on Zika today. The nation's top health officials warning that the Zika virus appears to be a lot scarier than first thought.
A stark warning that the haunting images of babies born with microcephaly in Brazil, abnormally small heads and brains, are becoming a very real concern in the U.S.
Birth defects caused by Zika virus make it a terrifying disease. But it pales in comparison to a far worse infection that plagued us through much of the 20th century.
Many of us think of German measles as just another one of those harmless childhood diseases, and that is true, it can be. But it also can be, as polio was, one of mankind's worst cripplers.
A severe rubella epidemic swept the United States in 1964-65, leaving behind literally thousands of damaged babies whose mothers had acquired rubella during pregnancy.
Although rubella was a mild infection of children, it wasn't mild at all if it infected a pregnant woman during her first trimester. Then she could go on to deliver babies who had severe congenital abnormalities, abnormalities for the eye, blindness, the ear, deafness, the heart, causing terrible abnormalities of the heart structure. And ironically, it's a virus that, when it infected women in the first trimester, could cause autism.
In fact, in 1964 and 65, rubella was so feared that approximately 5,000 U.S. women infected with the virus chose to surgically abort their pregnancies. And 6,000 more lost fetuses to spontaneous abortions. At the time, Dr. Stanley Plotkin was working at the Wistar Institute in Philadelphia.
As part of my work at Wistar and also the Children's Hospital, I was seeing both mothers who were concerned about whether to have abortions because of rubella in them and the possibility of intrauterine rubella in their infants, and also diagnosing the congenital syndrome. Those are experiences that certainly mark one and give impetus to trying to prevent things like that from ever happening again.
Plotkin would develop a rubella vaccine to protect a new generation. His first order of business? To acquire a strain of the virus.
The virus was isolated from one of the fetuses that were aborted at the mother's request because she had acquired rubella. And indeed, when we obtained the fetus, it was clear that the virus had damaged the fetus.
Dr. Plotkin's lab alone received material from roughly 35 pregnancies terminated because of rubella infection. His RA273 strain of rubella virus came from the third organ, the lungs, of the 27th damaged fetus he had received during the 1964-65 outbreak. To make his vaccine, Plotkin would need cells in which to grow the virus. Cells are like tiny factories, making proteins and enzymes needed by the cell and by the body. A genetic code, or DNA, provides the blueprint, instructing the cell's components on what to do. Viruses also contain a genetic code, but they can't make their own proteins, so they need to hijack the protein-making machinery of a cell to reproduce. Once inside a cell, the virus releases its genetic code, which takes over the protein-making machinery. The cell follows these new instructions and becomes an assembly line, generating thousands and thousands more virus particles. These new virus particles are an exact copy of the original virus, and soon leave the cell so they can infect other cells. In the 1960s, researchers had typically used animal cells to grow viruses for vaccines. But the head of vaccine development at Merck, Maurice Hilleman, had recently discovered something frightening about animal cells while working to improve Jonas Salk's polio vaccine.
What Dr. Hillman found was that the monkey kidney cells that were used both by Jonas Salk when he made his vaccine or Albert Sabin when he made his vaccine were contaminated with a monkey virus. It was the 40th known monkey virus of man. It was called SV40, and he found out soon enough that it also caused cancer in experimental animals.
This made people realize that cells from animals, as primary cells from animals, could be contaminated with a variety of so-called extraneous agents, that is, viruses that you don't want in a vaccine.
The challenge for Plotkin would be to find a type of cell that didn't carry the risk of contaminating viruses. It so happened that across the hall at the Wistar Institute, Dr. Leonard Hayflick had been experimenting to better understand how and why we age. He did it using human fetal cells.
Human diploid fibroblast cells are clean. That is to say, coming from fetal tissue, they have no agents that you don't want in your vaccine. So I decided that in order to avoid any problems of so-called extraneous agents, that I would use human cells for the attenuation.
Hayflick had received his fetal cells from Sven Gard at the Karolinska Institute in Sweden, the organization responsible for the Nobel Prize. Gard had obtained the fetal tissue from an elective abortion performed in the early 1960s, a fact that would later cause controversy. Because he was at the Wistar Institute at the time, Hayflick named his line of cells WI-38 cells.
A cell bank had been established at Wistar so that one didn't have to cultivate any new cells, but one could go back to the freezer, take out an ampule of the cells, grow them up, and then make a vaccine lot in those cells. I started to use my experience with polio vaccine, actually, to attenuate the virus, meaning, of course, to weaken it to the point where it would still immunize but would no longer cause disease.
Weakening viruses by attenuation is based on the principle of selection. Like with evolution, survival of the fittest is dictated by one's environment. Change the environment, say from the human body to human cells in a petri dish, and you suddenly change the whole game. Now, if you lower the temperature of the petri dish below that of the human body, all bets are off. The vast majority of virus particles will simply cease to reproduce, but a few will find they can adapt to this new environment. By passing these select virus particles over and over through the laboratory cultures, the virus population is soon made up of only those particles that can grow well in the petri dish, but can't grow well in the human body. Plotkin's temperature adaptation in fetal cells proved to be an efficient and effective means of attenuating his vaccine, and the work showed great promise. But not everyone thought so.
This was a new concept, and there was great difficulty in getting the regulatory authorities to accept the idea of using those cells to make vaccines. The attitude was, better the devil we know than the devil we don't know.
Plotkin couldn't have predicted just how much resistance his use of fetal cells would face, not only from the regulatory authorities, but also from his fellow scientists.
At the time, there was a dogma that human cells in particular could not be passed numerous times, or if they did, they would transform to cancerous type cells. But Hayflick and Moorhead showed that indeed the human cells would replicate, would multiply a limited number of times, and then they would literally senesce, that is, they would grow old, and the cultures would stop, would in effect die.
A meeting about rubella vaccines convened at the National Institutes of Health, and Plotkin was part of a minority advocating for the use of human fetal cells. Among those against the idea was oral polio vaccine developer Albert Sabin.
He got up towards the end of the meeting and made a statement about why diploid cells should not be used. But he had no evidence, and I remember very clearly saying to myself the biblical phrase, "The Lord has delivered him into my hands."
Plotkin addressed the meeting, pointing out that Sabin's statements were not based in fact, and that all the evidence showed fetal cells were indeed safe.
The audience gave me an ovation. Now, that was not for me personally, but simply because scientists in general don't like ex cathedra statements, and it was clear that most of them had been convinced that the prejudice against diploid cells was not founded.
If his scientific peers were beginning to come around, those in the religious community were still grappling with the morality of using human fetal cells, even when developing a life-saving vaccine.
Indeed, some controversy did arise because the cells that were used to grow the virus were obtained from a normal fetus.
This fact raised questions among anti-abortion advocates and ultimately drew the attention of the Pontifical Academy for Life, then headed by Cardinal Joseph Ratzinger, who would later become Pope Benedict XVI. While the Academy weighed the heavy toll of using cells from an elective abortion, Ratzinger ultimately cleared parishioners to use vaccines developed with fetal cells to avoid the devastating dangers to human health, especially from rubella.
Their consensus view is that as the cells were not derived specifically to develop the vaccine, and inasmuch as one is not doing new abortions for every vaccine lot, one is going back to cells that were frozen in the early 1960s, so that no immoral act is taking place according to those theologians. Now, I am sure there are other extreme points of view, and from my point of view, personal point of view, having seen the suffering of families with congenital rubella syndrome infants, I consider that what was done is 100% moral.
The specter of another rubella epidemic was looming, and Plotkin understood he wasn't the only one racing to head it off. Two other formidable forces were also hard at work: the U.S. government and the greatest vaccine innovator of all time, Maurice Hilleman.
The NIH at the same time was propagating the virus in bovine kidney for purpose of attenuation.
Hilleman found the NIH strain wasn't weak enough.
So I said, well, I guess maybe I should put it through ducts and attenuate it, which I did. Now we had the two viruses. So we used what was called the HPV 77 duck, and that became the vaccine.
By 1969, Plotkin had finalized his work on the RA273 strain of rubella vaccine, which was poised to prevent the next impending outbreak. But the U.S. regulatory authorities continued to question his use of human fetal cells. With the resources of Merck behind him, Hilleman's duck cell rubella vaccine was licensed in the U.S. that year. Plotkin's was passed over.
The British had a different attitude about human diploid cells from early on. That is, they were convinced that the objections to diploid cells were nonsense. And so they took up the diploid strain and started using it in Europe, continental Europe and the UK, at about the same time as rubella vaccination started in this country.
With Hilleman's vaccine working in the U.S. and Plotkin's in the UK, the rubella outbreaks expected in the early 1970s never happened. 20,000 cases of congenital rubella syndrome were avoided, 5,000 surgical abortions prevented, and 6,000 additional spontaneous abortions diverted. Vaccines had again removed a terror from our midst. In the U.S., Hilleman's rubella vaccine was working, and the science behind Plotkin's use of fetal cells was slowly gaining acceptance. Throughout the 1970s, the safety and effectiveness of each vaccine was closely watched.
It became clear that the immunity that was produced by the various strains was different. And at a certain point, Maurice Hilleman came to me and said, we think that the RA273 strain is better than what we're using, and we would like to produce it.
For Maurice, it was always about the science. It was always about the data. If there were data showing that Dr. Plotkin's vaccine was a better vaccine than his, then fine, we'll use Dr. Plotkin's vaccine.
The ironic thing is that human diploid cells are now considered to be the gold standard of the acceptable cell substrate. And there again, one sees the evolution of science. What was a heresy at the beginning is now the orthodoxy.
In 1979, Plotkin's RA273 rubella vaccine was licensed for use in the U.S. By 2005, rubella was eliminated from the United States completely. Today, fetal cells are also used to make hepatitis A vaccine, the chickenpox vaccine, and a vaccine against rabies, one of humankind's most feared diseases.
As a physician, unquestionably, the desire to prevent disease, even more than to cure disease, is a driving force. And I have to admit that the elimination of rubella from the United States, the elimination of rubella from the Western Hemisphere, is something that pleases me greatly. So, yes, there's satisfaction, but there's always more to be done. And I maintain enthusiasm because I see progress. And but even if I didn't, I think I would still want to try to move things forward.